Hello, welcome to the Bioinformatics Coach channel. In today's video, I'm going to give you 6 questions you need to ask and answer before analyzing bioinformatics data. So let's begin. And I assume that you have your data sets you want to analyze, and you also have your questions you want to answer by analyzing the data. So we begin. Question number 1. What kind of data am I dealing with? Are you dealing with transcriptome data? Are you dealing with proteomics data, whole genome sequence data, whole exome sequence data, etc.? You need to be able to find out, because the kind of data you have will determine what kinds of analysis you can do and what questions you can answer. Let's say, for example, you've been given transcriptome data. Let's say RNA-seq data. Then one of the things you can do is to be able to identify differentially expressed genes. And this is something that will be quite difficult to do if you are dealing with whole genome sequence data. So this is just an example of where the kind of data determines what questions you can answer and what information you can obtain at the end of the day. So it's important you get this question answered. This leads me to question number 2. What is the format of the data I'm dealing with? Are you dealing with FASTQ files? Are you dealing with VCF files? Are you dealing with FASTA files? Are you dealing with GenBank files, etc.? It's important because the format of the data will determine the software to use, and it will also determine where in the analysis pipeline you have to begin your study from. So let's say, for example, you have a study where you are looking at identifying genes, some genes present in a particular bacterial isolate, and then you've been given FASTQ files. FASTQ files are raw reads, and this means that you have to begin from doing some QC on the data, doing some trimming, performing genome assembly, polishing the assembly contigs, performing genome annotation, and from the annotation, you'll be able to identify your genes of interest. Okay, let's say for the same study, you've been given GenBank files. GenBank files contain annotation, and so this means that probably genome annotation has been done, and therefore you don't have to go back and start from scratch from trimming, QC, genome assembly and all that. You just take the GenBank file, start the analysis and identify the genes of interest. And because besides with GenBank files, you can't use GenBank files to perform genome assembly, so it makes sense about the format of the data, because they will determine the software to use and where to start the analysis from. This leads me to question number 3. What is the quality of the data? Quality of data is important because at the end of the day, you want your results to be 1, reproducible, 2, have a higher statistical power. So these things in some cases are determined by the quality of the data. So it's important that you get this information about quality. And talking about quality, if you are dealing with sequence data, then you could use tools like FastQC, MultiQC to assess the quality of the data, and this in turn will help you and determine what steps to take going forward. You could also improve the quality of the genome reads by doing trimming. So trimming with tools like Circle, Trimmomatic will help to remove contaminants, will help to remove adapters and reads that are of low quality, and these things in general will also help to improve the quality of the data. This leads me to question number 4. Do I have the skills and expertise to analyze the data? You are going to analyze bioinformatics data, and therefore you need to have some programming skills. You need to be able to write some bash scripts to handle the data and process the data. And these things are important before you can analyze the bioinformatics data. So if you have the skills and expertise, then it's great. You can start the analysis. But if you don't have the skills and expertise, then you need to look at where to get them. So talking about where to get the skills and expertise, you could enroll in bioinformatics courses, workshops, online and in person, where you are given information, you are being trained, and the materials you get, you practice, and then you gradually build the skills and expertise. You could also consider looking at collaborations where you collaborate with people who have the skills and expertise, and then these people will help you to analyze the data. You could also consider, if time is of essence, you could also consider contacting companies that do data analysis. Now we have companies that can analyze data, so you pay a fee and then they will analyze the data for you, and then you can just and get your results out and then publish your papers or whatever you want to do. You shouldn't also forget you can read publications and then papers to also try and then get some information about bioinformatics analysis and acquire some skills there as well. So it's important, this question of skills and expertise, whether they are available or not, you try to answer it. And this leads me to question number 5. Do I have the computing infrastructure to analyze the data? Talking about computing infrastructure, I'm referring to software, hardware, storage. Let's look at software, for example. You are going to analyze data, and therefore you need software to analyze. So the question is, are there software available to perform the analysis? Let's even say you want to perform genome assembly. Are there software available to help you do that? If the softwares are available, are they free? Or do I need to pay a fee? Or do I need to subscribe and then make some monthly payments? You need to be able to answer this, because it boils down to your analysis and the results, and therefore issue of software is very, very important. One good thing about bioinformatics is that a lot of the software that are being used are free. Just go online, download, install, and then start using them, and then you'll be fine. So generally speaking, you'll be fine with open source tools. But of course, if you have the money, if you have the funds, then you could also look at some proprietary software as well. So this leads me to number 2, hardware. With bioinformatics data is large, huge, complex, high dimensional, and therefore it becomes a challenge if you are going to use your normal laptops or everyday laptops to analyze the data. It's become very challenging. I remember I crashed one of my laptops some time ago because I was using it to analyze data that it wasn't supposed to be done. So you need to have some kind of high-end PCs and high-performing PCs that will help you to crunch the data. And therefore, this means you might consider using high-performance computing platforms that have large RAMs, that have large numbers of processors to help you to process and analyze your data quickly. You could also consider collaborating with institutions that have these high-performance computing platforms, and then they can grant you access, and then you can use them for your analysis. That is if you don't have the infrastructure locally in your institution. So it's something you might also want to think about. You could also contact cloud services. Now there are cloud services such as Google and Amazon, which can provide the hardware. They can provide the needed hardware for you to analyze the data. So you just pay a fee and then you start your analysis. And some too might let you use the platforms for free for a period. And then once the period is over, then you could consider paying and then continue using the services. So this issue of hardware is also important. This leads me to number 3, storage. With bioinformatics data, it's large. It can take quite a significant amount of storage space. So you need to do a proper planning with respect to storage when you are going to analyze bioinformatics data. Some of the data sets may be compressed, and therefore they may need to be decompressed before analyzing. And this means that when they are decompressed, they will occupy more storage than their compressed form. And so you need to be able to factor these things when you are looking at storage for your data. And some of the software also generates intermediate files, and these files, in addition to the original files, input files, will also occupy space. So all these things should be considered when planning for storage. So it's important you get this answered. So this leads me to the last but not the least. Are there workflows and pipelines available to analyze the data? You have to answer this. At the end of the day, in your projects, there are aims, there are questions you want to answer, and therefore you need to follow some protocols. You need to have some processes going on. There will be some activities you will do. Let's say, for example, bacterial genome analysis. You need to perform QC. You need to do some trimming, genome assembly, polishing, genome annotation, all that. These are processes, and each of these stages require a software. So what happens is that this software, you need to find a way to link them so that once one process is done, it is moved or it is pushed to another process, another software to continue the processing. And all these things leads to what I'm referring to as the workflows and pipeline, because there's that flow and there's that systematic processing. So workflows are important, and therefore you need to ask whether the workflows are available or not. If they are available, are they free, or do I need to pay a fee? If you don't have the workflows, you might consider building one from scratch. And if the workflows are also available, you try and then get them. Now, thanks to GitHub, there are lots and lots of bioinformatics and data analysis workflows and pipelines available. So all that you need to do is just go clone the repository and then start using the analysis, start doing the analysis using the cloned pipelines you have. So these are things you need to look at. Now, some of these cloud services also provide workflows and pipelines. GATK, Broad Institute, I think they also have some genome analysis pipelines or cloud services that you can actually use and then just get your data analysis done for you without having to build the pipelines from scratch. So it's important you also look at all these things before analyzing the bioinformatics data. So let me also get your views in the comment section. Let's discuss this and then let's get some stuff going. So that'll be all for this tutorial, and I'll see you in the next session. Goodbye.